Good morning everyone. Today we discuss locomotion and movement, new chapter. So what is locomotion? Movement of animal from one place to another place to obtain food, shelter, mate, known as locomotion. Movement is the unique character of organisms. All organisms exhibit movement, plants and animals. Plants also, due to wind current, the branches swing from one end to another end. So that is the movement. Or during physical exercise also, we may lift our hands, legs, or we rotate our head or bend. So our body. So these are different movements. So due to movements, no change in the place of the individual. So no displacement of the body due to movement. But due to some movements, displacement of the body also take place. For example, walking, swimming, running, climbing. So these are also movements, but these movements change the displacement, change the place of the organism. So the movements change the place of organism, lead to locomotion. So all movements are not locomotion, all locomotions are movements only. All movements never displace the body, change the place of the organisms. So movement of animal from one place to another place to obtain food, shelter, mate, or escape from the predator is known as locomotion. Movement is the unique character of organism. Both plants and animals exhibit movement. During physical exercise also, we move our body parts, but no change in the place of the organism. Due to some movements, a change occur in the place of the organism, that is known as locomotion. So due to some movements, change occur in the displacement of the body. It is known as locomotion. So all movements are not locomotion. All locomotions are movements only. Movement with the help of cilia, known as ciliary movement. Movement with the help of pseudopodia. Pseudopodia, amoeboid movement. Amoeboid movement. Movement with the help of flagella, flagellar movement. Flagellar movement. Pseudopodia movement seen in protozoans like amoeba. It is the pseudopodium. False foot. Pseudopodium means false foot. Amoeba moves in the direction of formation of pseudopodia. These are the pseudopodia in forward direction. These are the pseudopodia present in the rearing end, posterior end. The pseudopodia present at the posterior end disappear. New pseudopodia are formed from the anterior end. The amoeba moves in the direction of formation of pseudopodia. Pseudo means false, podia means foot, false foot. So pseudopodia movement with the help of pseudopodia known as amoeboid movement. Pseudopodia help in food capture and locomotion. Cilia, for example, paramecium. On the surface of the body, cilia, short hair-like structures are present. Paramecium. The protozoan contains cilia known as ciliate. So the cilia help in locomotion. The cilia present in the oral groove help in food capture. So example paramecium. Next one, locomotion by flagella is known as flagellar locomotion. Example euglena. Locomotion by flagella. A long hunter-like structure, flagellum, is known as flagellar locomotion. Flagellar locomotion. So flagella help in only locomotion, not help in food capture. Pseudopodia and cilia help in both locomotion and food capture. Flagella help in only locomotion. The flagellum push the water in backward direction. The backwardly directed water push the body forward direction. Here also cilia push the water backwardly. The backwardly directed water push the body forward direction. So similarly tentacles. Tentacle, also known as arm. The tentacle push the water backward direction. Animal moves forward direction. The tentacles also help in food capture. The tentacle capture the food. Later the tentacles contract. Now the prey is nearer to the mouth. Now the animal engulf the food. So in this manner, movement with the help of tentacular movement. Tentacles also help in food capture. So tentacle perform both locomotion and food capture. So these are the various locomotory organelles and organs. Cilia in paramecium, flagella in euglena, pseudopodia in amoeba, tentacles for example in hydra. Tentacles, pseudopodia, cilia perform both locomotion and food capture. Flagella perform only locomotion, or flagella help in only locomotion. So in our body, amoeboid movement, ciliary movement, our body also exhibit amoeboid movement, ciliary movement, flagellar movement, flagellar movement. Amoeboid movement means with the help of pseudopodia. Macrophages, in the textbook given macrophages and leukocytes. What are macrophages? Monocytes present in the connective tissue known as macrophages. All leukocytes acquire pseudopodia. With the help of pseudopodia they perform locomotion. The monocytes present in the connective tissue known as macrophages. So they develop pseudopodia and perform locomotion with pseudopodia known as amoeboid movement. So in our body, macrophages and leukocytes means white blood corpuscles. Macrophages, monocytes present in the connective tissue. They are also one type of leukocytes. Perform amoeboid movement. ciliary movement. Trachea, inner surface of trachea, windpipe contains cilia. Similarly, fallopian tube, oviduct in female known as fallopian tube contains cilia. All cilia strike in one direction. Any solids present in trachea from the trachea enter into larynx, from larynx into pharynx, from pharynx we spit it out. So our respiratory system does not allow any solids. Similarly, fallopian tubule, oviduct also known as fallopian tubule, lined by cilia. The cilia present in fallopian tubule help in transport of ova. So, inner ducts, in our body, inner ducts lined by cilia. For example, trachea help in send out the solids from respiratory route. Similarly, cilia present in fallopian tubule help in the movement of ova. Flagellar movement only in our body spermatozoa. That means only male human being. Flagellar movement. So, flagellar movement by spermatozoa. Spermatozoa male gametes. They perform flagellar movement in male human beings and female human beings also. Why? The same spermatozoa enter into fallopian tube and perform flagellar locomotion. So, flagellar movement exhibited by spermatozoa. Ciliary movement in trachea and fallopian tubule. Amoeboid movement by macrophages and leukocytes. Macrophages also one type of leukocytes only. Leuco means white, cyte means cell. White blood corpuscles. Muscular movement. Movement with the help of muscles known as muscular movement. Movement of jaws, movement of hand, legs, locomotion, all due to involvement of muscles, particularly striated muscles. Muscular movement, muscles responsible for muscular movement. Muscles are about 40 to 50% of total body weight. Muscles are about 40 to 50% of total body weight. Now we discuss striated muscle. So striated muscles are under the control of will of organism. So the striated muscles responsible for locomotion, movement of various organs. So striated muscles are under the control of will of organism. Muscles present in visceral organs known as visceral muscles. What are viscera? Organs present in the coelom known as viscera. So muscles present in the viscera are smooth muscles. Muscles responsible for locomotion, striated muscles. Striated muscles mainly present in the limbs, forelimbs, hind limbs. So before we discuss muscular movement, we discuss the structure of striated muscle. Striated muscle cell. So muscle, whatever may be the type of muscle contain, we already discussed while discussing tissues. Once again I revise. Whatever may be the type of muscle contains several fibers parallel to the longitudinal axis. These fibers are known as muscle cells or muscle fibers. Each muscle fiber contains several fibers parallel to the longitudinal axis. They are known as myofibrils. Each myofibril contains two types of myofilaments. Myosin and actin myofilaments present alternately in striated myofibril and cardiac myofibril. In case of smooth myofibril, myosin and actin myofilaments present side by side. In smooth, for example, myofibril of smooth muscle cell in myofibril of smooth muscle cell, myosin and actin filaments are present side by side. That's why striations, lines not appear. In striated myofibril and cardiac myofibril, myofilaments are arranged alternately, myosin myofilaments and actin myofilaments. The part of the myofibril contain only myosin filaments known as A band. The part of the myofibril generally contain only actin myofilaments known as I band. A band and I band arranged alternately. So once again, whatever may be the muscle, each muscle contains several fibers parallel to the longitudinal axis known as muscle cell. Muscle cell also known as muscle fiber. Each muscle fiber contains several fibers parallel to the longitudinal axis known as myofibril. Each myofibril, in case of striated muscle cell and cardiac muscle cell, two types of myofilaments are present alternately. Myosin myofilaments, actin myofilaments, myosin actin. In this manner, they present alternately. But in the myofibril of smooth muscle cell, myosin and actin filaments are present side by side. Now we discuss the structure of striated muscle. The plasma membrane of striated muscle is known as sarcolemma. Cytoplasm of the striated muscle cell known as sarcoplasmic reticulum. Just below the sarcolemma, several nuclei are present. So one cell contains several nuclei, multinucleate state. It is known as syncytial state. If an animal cell contains several nuclei, this condition is known as syncytial state. If a plant cell contains several nuclei, known as sinusotic. Why one muscle cell contains several nuclei? In embryonic stage, several myoblast cells unite. As a result, one striated muscle cell is formed. So the cells unite, but nuclei never fuses. That's why several nuclei present just below the sarcolemma. In the sarcoplasmic reticulum, numerous myofibrils are present, but I drew only 3. In all myofibrils, in a particular position, myofibrils coincide and appear as line. So like this. Appear as a line. You already know the part of the myofibril contain myosin filaments is known as A band. So number of lines depends upon number of A bands. 1, 2, 3, 4. That means 4 lines appear in the striated muscle cell. So striation means lines. Why the muscle cell got the name striated? All myofibrils present, all myosin myofilaments present in all myofibrils at a particular position coincide and appear as a line. Again A bands, another line, A bands, another line. So, in a striated muscle cell, numerous myofibrils are present one end to another end. In all the myofibrils, myosin filaments coincide and appear as a line. That's why it got the name striated. Striations also seen in cardiac muscle. Striations absent in smooth muscle cell. We already discussed in smooth muscle cell, myosin and actin filaments are present side by side. Mitochondria of the muscle cell known as sarcosome. Endoplasmic reticulum is known as sarcoplasmic reticulum. You already know striated muscles are attached to skeleton. That's why striated muscles also known as skeletal muscle. The striated muscles undergo rapid contraction relaxation. That's why they undergo fatigue. Fatigue means great tiredness. They undergo fatigue, great tiredness. So only striated muscles undergo fatigue. That's why they required rest. After taking rest only, again they undergo contraction and relaxation. So this is about striated muscle cell. Striated muscle cell. So every muscle contains several fibers, parallel muscle cell. Each muscle cell contains parallelly several fibers longitudinally. Parallel to the longitudinal axis known as myofibril. Each myofibril contains two types of filaments, myofilaments, myosin myofilaments, actin myofilaments. So structure of a striated muscle cell. The plasma membrane of the muscle cell, sarcolemma. Cytoplasm of the muscle cell, sarcoplasmic reticulum. Just below the sarcolemma, several nuclei are present. So syncytial condition. Syncytium means multinucleate state. Why striated muscle cell contains several nuclei? In embryonic stage, one striated muscle cell formed by the union of several myoblast cells. Mitochondria of the muscle cell, sarcosome. Endoplasmic reticulum of the muscle cell, sarcoplasmic reticulum. Sarcoplasmic reticulum contains abundant number of cisternae. So the cisternae store the energy in the form of ATP. And sarcoplasmic reticulum contains cisternae. Cisternae contain calcium ions. So check this point. Not ATP. ATP present in the mitochondria. Endoplasmic reticulum of the muscle cell is known as sarcoplasmic reticulum. Sarcoplasmic reticulum contains cisternae. Cisternae store abundant number of calcium ions. Calcium ions are responsible for muscle contraction. ATP molecules present in the sarcosome. Mitochondria of the muscle cell is known as sarcosome. So readily usable energy. So that is ATP molecules present in the sarcosome. So why striations upper? Striation means lines. In all myofibrils, myosin filaments present in a particular area coincide, appear as a line. That's why these muscle cells got the name striation. Striated means line. Striations appear in the striated muscle cell and cardiac muscle cell. Why striations not appear in smooth muscle cell? In smooth muscle cell, myosin and actin myofilaments are arranged alternately. That's why striations not seen in smooth muscle cell. Now we discuss the structure of myofibril. Now we discuss the structure of myofibril. Actually we discuss the structure of striated muscle cell while discussing muscular tissue. Muscular tissue is derived from mesoderm. Muscles are about 40 to 50% of total body weight. Structure of a striated myofibril. Striated myofibril. Myofibril contain myosin myofilaments and actin myofilaments alternately. The part of the myofibril contain mainly myosin filaments known as A band or anisotropic band. Light rays are passing through myosin myofilaments, that's why it is dark. The part of the myofibril contain only actin filaments known as I band or isotropic band. Light rays are reflected, that's why it is bright band. The dark membrane present in the middle region of A band known as M line. The dark membrane present in the middle region of I band known as Z layer or Cruces membrane or Debye line. So in this manner, A bands and I bands present alternately from one end to another end of the striated myofibril. So striated myofibril contain myosin myofilaments and actin myofilaments alternately from anterior to posterior part of the myofibril. The part of the myofibril mainly contain myosin filaments is known as A band or anisotropic band. Anisotropic band also known as A band or anisotropic band. It is dark. Why A band is dark? Light rays are passing through A band, that's why it is dark. The part of the myofibril contain only actin filaments known as I band or isotropic band. Light rays are reflected, that's why it is bright. In this manner, A band and I band present alternately from one end of the myofibril to another end. The sarcolemma of A band, in the middle region of A band, sarcolemma extend from one end to another end like a thin membrane. This thin membrane is known as M line. Similarly, in the I band, the sarcolemma extend from one end to another end, that is known as Z layer or Cruces membrane. Z layer also known as Cruces membrane, also known as Debye line. Cruces membrane or Debye line. The portion of the myofibril between two successive Z layers is known as sarcomere. Sarcomere is the functional unit of muscle. What is sarcomere? The portion of the myofibril between two successive Z layer, it is one Z layer and it is a successive next Z layer. The portion of the myofibril between two successive Z layers is known as sarcomere. Sarcomere is the functional unit of muscle. In a sarcomere, complete A band, above the A band, half of the I band, below the A band, half of the I band. So sarcomere contain A band and I band, but they are not present alternately. A band, complete A band is present. Above the A band, half of the I band. Below the A band, half of the I band. Sarcomere. A band contain peripheral part, so it is the A band. Peripheral part of A band contain both actin and myosin filaments. Observe, it is A band. Peripheral means sides. Peripheral part of A band contain both myosin and actin. So another peripheral part also contain actin and myosin. So in A band only myosin filaments are present, wrong. In A band only actin filaments are present, wrong. So in relaxed state, so it is the sarcomere in relaxed state. In resting condition, the peripheral part of A band, it is one peripheral part, it is another peripheral part. Peripheral part contain both actin and myosin filaments. What is H zone? The part of the A band contain only myosin filaments. Observe, only myosin filament. The part of the A band contain only myosin filament is known as H zone. I band contain only actin filaments. So H zone is lighter. when compared to peripheral part of A band. Why H zone contain only myosin filaments? Peripheral part of A band contain both myosin and actin. That's why the darkest part of the sarcomere is peripheral part of A band. Second one is H zone. What is H zone? Lighter zone present in the A band is known as H zone. Why H zone is lighter in the A band? Due to presence of only myosin filaments. Peripheral part of A band contain both myosin and actin, but the central part of A band containing only myosin. Due to presence of myosin filament, H zone is lighter when compared to peripheral part of A band. So simple definition, lighter zone of A band is known as H zone. H zone contain only myosin filament. Peripheral part of A band contain both actin and myosin filament. I band contain only actin filament. So due to presence of only actin filaments, I band is the thinnest region. H zone is thick when compared to I band, thin when compared to peripheral part of A band. Sometimes they may ask, arrange the following parts of myofibril, striated myofibril in increasing order or decreasing order of thickness. In increasing order of thickness, I band. Next H zone, third peripheral part of A band. I band is the thinnest region due to presence of only actin filaments. Actin filaments also known as thin filaments. H zone contain only thick filaments. That's why H zone is thick when compared to I band, thin when compared to peripheral part of A band. Why? Peripheral part contain both thin and thick. Due to presence of both thin and thick, peripheral part of A band is the thickest part. Due to presence of only myosin filament, H zone is thin when compared to peripheral part of A band, thick when compared to I band. So structure of striated myofibril. In a myofibril, myosin filaments and actin filaments are arranged alternately. The part of the myofibril mainly contain myosin filaments is known as A band or anisotropic band. So light rays are passing through A band, that's why it is dark. The part of the myosin filament contain only actin filaments known as I band or isotropic band. Light rays are reflected, that's why it is bright. Dark band is A band, bright band is I band. Next point, the lighter membrane or the lighter membrane present in the middle region of A band is known as M line. How M line is formed? Extension of sarcolemma from one end to another end, M line. The lighter membrane present in the middle region of I band known as Z layer or Crassus membrane or Debois line. The portion of the myofibril between two successive Z layers known as sarcomere. Sarcomere is the functional unit of muscle. Sarcomere contain complete A band, above the A band, half of the I band, below the A band, half of the I band. Sarcomere. Sarcomere is the functional unit of muscle. Sarcomere is the functional unit of muscle. Peripheral part of A band contain both myosin and actin filaments. That's why in the sarcomere, peripheral part of A band is the thickest part. Middle part of the A band contain only myosin filament. The part of the A band contain only myosin filaments known as H zone. The part of the sarcomere contain only actin filaments, so that is the I band. So I band is the thinnest region. H zone is thick when compared to I band, thin when compared to peripheral part of A band. Structure of sarcomere. So sarcomere contain complete A band, above the A band, half of the I band, below the A band, half of the I band. Now we discuss the sarcomere given in the textbook. It is the actual diagram, but we follow the textbook. So we discuss the sarcomere diagram given in the textbook. Look on the board. It is a sarcomere, A band. It is the A band. It is half of the A band. It is another half. It is Z layer. It is another Z layer. You already know the portion of the myofibril between two successive Z layer is known as sarcomere. Z layer, Z layer, Z layer, Z layer. So while observing under microscope, the membrane present in the middle region of I band appear as alphabet Z. That's why it got the name Z layer or deboiline or Crassus membrane. So the portion of myofibril from one Z layer to successive Z layer is known as sarcomere. Sarcomere contain half of the I band, complete A band. So half of the I band left to A band and half of the I band right to A band. So sarcomere contain half of the I band left side, left to half of the I band, right side complete A band, right to complete A band, half of the I band. So sarcomere contain complete A band, half of the I band left side, half of the I band right side. I band contain only actin filaments. A band contain both actin and myosin filaments. The part of the A band contain both actin and myosin known as peripheral part. So the peripheral part of A band is the thickest part of sarcomere. The middle part of A band is lighter. The lighter part of A band is known as H zone. Why the middle part of A band is lighter when compared to peripheral part? The middle part of A band contain only myosin filaments. Due to presence of only myosin filaments, the middle part of A band is lighter. It is known as H zone. I band contain only actin filament. So I band contain only actin filaments. Peripheral part of A band contain both myosin and actin filaments. H zone contain only myosin filaments. So due to presence of both actin and myosin filaments, peripheral part of A band is the thickest zone. H zone is lighter when compared to peripheral part of A band and thick when compared to I band. So sarcomere. The dark line present in the middle region of A band is known as M line. Structure of sarcomere given in the textbook. So tomorrow we discuss proteins present in the filaments. Before we discuss the muscle contraction, few points regarding striated muscle. Later we discuss muscle contraction.